Is Jillian Pearring. I am an assistant professor at the University of Michigan. It's in Ann Arbor, Michigan, so about 45 minutes outside Detroit. And I have been here for about 6 years. So I started my lab in 2017. This is my first faculty position. I'm in the Department of Ophthalmology. So I actually sit in sort of a clinical building, and they have some research lab space on the top floor of that building. And so that's where I have my research group, where our labs are sort of housed and spaced and where we do most of our work. I also have a joint appointment in the Department of Cell and Developmental Biology. So that would be sort of more of a traditional graduate school program. Like ophthalmology is going to be more of a medical school program, right? Where you have med students and stuff, but in cell and developmental biology, you would have more traditional science kind of courses. And so that's a really nice sort of duality for me, where I can be sort of one foot in a sort of clinical environment, because a lot of my work translates into sort of human disease. But I can also have one foot in sort of the basic sciences, as we call them, where you study more of the sort of underlying mechanisms of how tissues and cells work and don't work, during disease. That's also where I get a lot of my sort of cohort of students and things like that who come through the lab to do research projects. I work with undergraduate students, high school students. I work with, graduate students, and then what we call postdoctoral fellows. So those are people who've actually received their PhD degree and then are doing additional research. It's sort of an academic research environment to sort of boost your knowledge and your skills. I myself did my postdoctoral research at Duke University down in Durham, North Carolina. So that was where I was before I was a faculty member here at the University of Michigan. In there, I was also in an ophthalmology department. So I sort of had done research in the vision science as a postdoc. So that's why I was sort of attracted to coming to Michigan and being in this ophthalmology department. I did my PhD degree at the University of Louisville in Louisville, Kentucky. And that was in biochemistry and molecular biology. And then I did my bachelor's degree at Boston University in Boston, Massachusetts. And then I was actually born and raised in a small town in Colorado called Manitou Springs.
Oh, yeah.
I have moved around a lot during my education. I think that's maybe a common theme for academic scientists. You sort of go to the school or the institution where you feel like you're going to get the best education or work with the best people, which means that you're oftentimes bouncing around every
Yeah.
handful of.
And then you mentioned being in like a lot of clinical settings. So how does your role differ from like an MD, PhD?
Very different. So if you have sort of the MD part of the degree, you're going to have a much heavier clinical sort of presence, right? So you will actually be seeing patients, you'll be handling caseloads like that. Maybe you're a retinal surgeon, or maybe you general practicer or something. Whatever your specialty is, you will actually have interaction with patients, which I don't do any of that. I'm strictly a PhD. I'm at the bench, as they say, right? So I'm sequestered up in the labs and I'm doing research all the time. So some of my colleagues are MD, PhD, and they have this sort of duality where they spend half their time with patients, and then they also spend half their time, in the research labs as well. I like being in this environment because I kind of have like a nice collaborations with people who do interact with patients. I get to sort of like hear a lot of the translational side of the work, but I am not an MD. I never studied medicine. I couldn't give you like an eye exam. None of that stuff. Like I am just a straight up scientist, right? Like I put on the science lab coat and the goggles and I go look in microscopes and I, do different models. I run a lot of assays, to look at different changes the cells have, or it's like different changes in protein levels or things like that. So that's the big, big difference between me and an MD, PhD.
Yeah. Thank you for the clarification. And
Yeah,
then do
yeah.
you mind talking a little bit more like in depth about your research, but in terms that are like understandable to a high schooler?
So my lab is very interested in studying photoreceptor cells. So photoreceptor cells are the light sensing cells that sit in your retina. So if you think about the eye, right, this is something that most people can have a good sense of, right? This is a big organ in our body, the eye, and it's involved in human vision. And light is coming through the front of your eye, right? This is where your cornea is, right? And where you have your lens. And that light is then diffracted from your lens, just like in a camera, sort of diffracted back onto this thin piece of neural tissue called the retina that's sitting at the very back of your eye. So if you think of your eye as sort of a globe, you have sort of one side is the front where you have the light coming in, and in the back, that light is being nicely positioned right onto this neural tissue called the retina. And this retinal tissue, if we take a cross section here, you can see is actually directly connected to the brain through this little connective axons here, the optic nerve. So this neural tissue is actually considered a piece of your brain that's sitting inside your eye. And it's responsible for detecting light and then taking that light and transforming it into an electrical signal that then can be transmitted to your brain so that you can then visualize the entire natural world, right? So you can actually see where we're going. And so if we just sort of zoom in a little bit to this retinal tissue, it has a lot of the similar hallmarks to other parts of your brain, right? What we know about the brain is it tends to be very layered, like neurons are talking to each other. So they kind of are put together in layers so they can make these connections and and then talk to each other through synapses. And it's the same way in the retina. You have these really nice layers. So here what we're seeing is this dense staining are the nuclei of one type of cell sitting in the retina. And here we have some a little bit lighter, but there is some density here too. This is a different type of cell sitting in the next layer. And these two cell types have to talk to each other. These are the light sensitive photoreceptors. And then these are some inner retina cells here. So the photoreceptors we think of as being sort of the very first cells detecting the light. These are the cells that are going to take all of those photons of light that are coming into your eye, and they're going to convert that into an electrical signal to send to your brain, right? So this is really, really important. This is sort of like, we call them the sensory neurons. They're the very first neurons to be sensing your environment, your light, right? You have similar neurons that are like in your nose, sensing odors. You have similar neurons in your mouth, sensing the different chemicals for taste. You have ones in your ears that are sensing sound waves, right? So these are what we call sort of globally sensory neurons. And in the retina, these are photoreceptor neurons here. And what's very cool about the photoreceptors, they actually are sitting at the very back of the eye. So the light actually goes all the way through these other neurons and all these other cell types here, and actually is precisely focused onto this very back layer called the outer segment layer here. You can see that there's like these like little like cylinders that are sort of stacked up next to each other, kind of like looking like little hairs that are all just kind of like situated next to each other. And these are actually individual cells. This is one compartment of an individual cell. And this compartment is specifically designed just to capture all of that light and convert that into electrical response. Right. So this is sort of what I like to call the business end, right? So this is where all the light is being captured. And actually the response is then electrical and chemical responses are then sent the other direction through the retina. So you take the light information and you then transmit it as a chemical response, first through electrical response and then chemical release down the other side of the cell. So this cell actually has to be built in a really special way where light information is being taken in from one side of the cell, and then chemical information is being released from the other side of the cell, and that they're talking to each other through this electrical change in the cell.
But that's really cool. I didn't know it was so complicated.
Oh, it's very complicated, but that's good for me, right? Because I like I like puzzles, and I like to sort of like think hard about how things are working in the cell, right? That's the thing that's a big part of being a scientist, is having sort of this inquisitive nature about, this is a complicated problem, and how can we try to understand how, things have been resolved in nature, how the cell has like been able to establish these very unique compartments within the cell to handle these different functions, right? And that's really the sort of like bulk of what my research group is investigating. And a reason that this is very important is because we see that in a lot of human disease where people are going blind over time, one of the common things that happen in the majority of these cases is that the photoreceptor cells are the very first cells to actually be lost. So this is actually a mouse model that we use, where you can see that all of these cells, which are the photoreceptors sitting in the mouse model, are now sort of gone at the same time point in this mouse model. So this is recapitulating a lot of human blindness that, a lot of my clinical friends have patient groups with, that they're working with these people, and we're trying to understand sort of the molecular mechanisms that are leading to this loss of this very important cell type. I think most high schoolers probably have heard of photoreceptors. Oftentimes they're called rod and cones,
Yeah.
and that is literally because of the shape of this light sensing compartment. As you can see here, I sort of called them as like little hairs or cylinders that are sitting there. Those are the rods. They're very long and thin, and that's why they were originally called a rod cell, because they had this very cylindrical rod shape to that compartment. And then the cones were actually a little bit more triangular, where it kind of like tapers off. They're a little bit harder to see because there's less of them in a mouse retina, but they're sort of sitting in here. I think there's one right here that's kind of smaller and thin here. And so these two flavors of photoreceptors have different functions and actually capture light under different conditions, as well as capture different wavelengths of light. And I could talk for an hour about that, but We don't have to do that, right? But I'm just trying to sort of point out that like some of the light information is being encoded right at this point. Like depending on which cell is going to respond to which particular light will give you information about color or about intensity, like the brightness levels that are in the room and things like that. So these are sort of important aspects of the photoreceptor. And in my cell, I mean, in my lab group, we are really interested in how that particular light compartment is sort of organizing itself and is getting all of the molecules that are needed to capture the light and transform it into this electrical response within that compartment specifically. And so here I'm just showing you some of the sort of images that we take in the lab where we're using a confocal microscope to actually label individual molecules within the tissue and then show their localization, right? And so here I'm labeling one of the molecules that is very important for establishing the outer segment compartment and involved in sort of phototransduction. And you can see that it's only located in this part of the cell, right? So it's only in these very nice cylinder sort of rod parts of the cell, whereas a different protein that's actually involved in releasing the chemicals from the other part of the cell is actually not allowed into that compartment, right? So there's this restrictive barrier that's sort of holding these two things as separate. And that's really important so that you're sort of putting molecules in the right place so they can actually do their function, right? And there's a lot of disease mechanisms where you'll have either one of these components displaced into the wrong compartment, and then the cell doesn't function properly. It doesn't know how to then sort of capturing light and making these sort of chemical changes. But yeah, so this is really like the bulk of the work that we do in the lab is just looking at how these two different compartments are sort of formed and then as well as maintained over the course of life. Because as with all neurons in your body, right? Once you produce them, that's the ones you have for the rest of your life. So once they're gone, you can't use more of them, right? So that's another aspect, is not just their formation, but also being able to sort of keep them intact and working over time as well. So I think that kind of encompasses the work that we do in the lab. But at heart, like I would say I'm a neuroscientist and I'm a cell biologist, right? So I sort of like to think about these neurons, these cells that are responding to light. And I like to think about how each of these cells individually is sort of organizing, all of the different molecules within them to do the right functions. That's sort of the gist of it. So
No, that's incredible. I feel like in most of my bio classes, we just kind of go over cones and rods like very briefly. And so I didn't know that I was this interesting.
it's incredibly interesting, I mean, when you just think about how many trillions of photons have to enter into your eye every minute, right, that are coming in, and that all of that information that you want to be able to capture and use so that we can sort of navigate our space, right? And we have very high resolution vision, right, as human beings, and being able to sort of discern all of the different colors, all of the different movements and all of that kind of stuff. I mean, really, this is the starting place for all of that information. And there's a lot of connectivity downstream of the photoreceptors that really then begins to piece apart gradients of color or the different places that things are moving within the scene, something that would be, within the center part of your vision versus something that's sort of peripheral to your vision. And all those kinds of things are really important.
And then my next few questions are a little bit more geared towards high schoolers.
Sure.
So how did you find your passion, and how do you suggest high schoolers like try to find theirs?
I would say that I always enjoyed my sort of math and science classes when I was in high school. I found those to be very interesting. I think I also had very good teachers. That's always a benefit when you have somebody who really can inspire you, and sort of give you a flavor of like, oh, this, of what it is to be a scientist or what it means to sort of do science. And I had a very excellent biology teacher in high school who convinced me to go to the local college in our town, Colorado College in Colorado Springs, and do a sort of a research intensive project during my junior year of high school. And so that really sort of was the first eye-opening experience I had for like what bench science looked like, like just sort of the day-to-day science of what it means to be a scientist, like sort of showing up, working with chemicals, working with different, what we call biochemistry. So whether it's with proteins or nucleic acids like DNA or RNA, and sort of like understanding how you can, manipulate and run different assays to sort of look at these different things that are happening. So I started pretty young in that world. I went as a high school student and did some making some agarose gels, and I ran a couple of PCR reactions, right? So this is where you're actually sort of like, taking the DNA content from something and then amplifying one particular part of the DNA, like a gene of interest, right? And you're just taking that one gene and you amplify it a whole lot so you can actually visualize it on a gel. right? Some of those simple things I started doing right in high school. So when I went to college, I still didn't really quite understand that you could just like do science as a career. Like I just sort of felt like choice was medical school. Like that was the sort of like thing that most people did with a science degree, was either engineering or medical school. And I just sort of didn't realize that you could be a scientist, right? Like that could be your
Yeah.
job. So I initially was pre-med, right? Thinking that that would be sort of the way that I would continue this love of sort of discovery. And I did a rotation during freshman year where I rotated in the ER. Did not enjoy it, basically. I'll just say I didn't enjoy that aspect of sort of the franticness of medicine. And also the sort of like, there's a lot you just don't know in medicine, and you're kind of throwing, the toolkit that you have, which is generally pharmaceuticals, at a problem or something like that, right? But
Yeah.
you're not really diving into understanding how something is working at this other level. And so I found that that was not resonating with me. The next year, I went to look for another lab experience, just thinking that, I really enjoyed that as a high school student, even if it's as simple as just, running assays all day. Like I just liked to sort of get down to that level. And I, again, just got really lucky with a good mentor. Again, I think that's sort of a common theme, is having, people who help to guide your hand as you make these decisions and think about these things critically. He was an excellent teacher, and he was also patient, and he also was really good at sort of lifting you up in a way that, even when things weren't working, you were enjoying yourself.
Yeah.
Because there's a lot of times that things aren't working, right? That made me realize that like I liked that kind of detective work, right? At the end of the day, that's what we're doing, right? Here's a hypothesis. How do we go about uncovering, elements of that hypothesis to figure out what's going on, right? So that's sort of the thing that I always really liked about it. And so then it was like, well, I should just go get a PhD. I should just not do the MD. But I think that, there's lots of other things that you can do in sciences too. And if, over time, I've had friends go and do it all, right? From, doing sort of more written stuff, being sort of a scientific writer, right? Where you're actually sort of doing more of a communications kind of work with your degree, whether you're communicating science to, general population, or communicating science to, doctors or nurses or something like that. There's a lot of people I know that have done those kinds of jobs, or more traditional sort of like teaching positions. I think science is cool, and it's an amazing thing. And, if you find yourself sort of asking more questions, I think that's a good sign that you should stick with science. If you like to ask questions because you want to know, right, how things are working, I think that that's always usually a good sign to be doing
Yeah.
science.
And then earlier you mentioned working with high school students. I imagine you get a lot of cold emails, like a lot of requests to join your lab. How do you pick out a student? Like what makes someone stand out to you?
Through the university policies, I'm not allowed to take cold email requests from high schooler students. They have to be through some sort of a program. So there's a number of programs here in the Ann Arbor area where the local high schools have sort of, I imagine it's something similar. You have like this research catalyst that you guys have or whatever, where
Like a feeder school, right?
exactly, where students who are particularly interested are like sort of being selected for these programs or signing up for these programs. And then we're working within those realms, because with minors, we're not allowed to just like have minors in the lab.
Yeah.
It kind of has to be sort of a pre-designed program that we're sort of working within. But with undergrads, it is just a cold email that they're all sending, and I do get a lot of them. And that is a lot harder to sort of suss out. There, a lot of it is really in how well you communicate your interests in that initial email, right? So at the beginning of a semester, I may receive 20 or 30 emails from interested undergrads that want to have some kind of a research experience, right? I mean, this is a big university. There's a lot of labs that they can go to for sure, and so in that, like being able to sort of really show that you're not just sort of massively like spam emailing every professor with the same blurb is really helpful. Like giving me a little bit of a sense that like, hey, I did look at your website and I am actually interested in photoreceptors. Like this is something that I could be like wanting to study, not just like, give me random opportunity because, that's what I think I want. But like it does help having a little bit of a couple of sentences in there just to sort of like say, hey, I took a little bit of extra time. I looked up some of this stuff and this would be interesting to me. It doesn't have to be much more than that, but I think those emails always sort of like rise to the top.
And then the last question is, if someone doesn't have access to a wet lab, what kind of skills do you think someone can still develop?
Yeah, there's lots of great skills. I mean, especially with science these days, it's really a lot about, big data is a big thing. So computer science skills are always highly valued. Being able to code, in R or doing any kind of like statistics, any of that kind of stuff. I mean, that always is like seen as a huge benefit for students coming in. Those are like mathematical skills at all. Like all that stuff really translates. I always say like ultimately, all the science is founded in math, right? Like it
Yeah.
all comes back down to some mathematical equation, right? So really keeping those things, if you can't get into a wet lab sort of situation, if you can do, courses on coding or you can do courses on statistics, even like something like websites and stuff like that, like because a lot of the stuff that we're doing does that kind of stuff. So I mean, all of that is all helpful. Like even just showing that you have sort of a creative tilt, I think always helps too. Like, hey, I'm creatively thinking about this, or I couldn't do this other thing, so I tried this and I thought that this might sort of like fill that gap, so just showing interest, I think, is a big part.
When I started doing research, I didn't realize how much failure it was and like how many go arounds you need to find to actually be able to get something done. Like I thought going in it'd just be straightforward, but there's a lot of bumps.
Yeah, I mean, that my graduate professor used to always say that's what research means. You're constantly redoing and redoing, constantly going back to the hypothesis and reassessing, right? And it can feel a little daunting at times, but I think like there, I mean, hold on to the highs. Like we had a big eureka moment with a student and I just sort of like dived in.
Congratulations.
Again, we was just like, wow, this is striking. This is amazing. Let's like think hard about what this means. And, I think that holding on to those highs is like really important, and recognizing that like sometimes the struggles or the sort of having to redo things, can lead you places you didn't expect, right? And that can also be really fun. So you have to just also sort of keep an open mind to what things mean and how a hypothesis can change, right?
Thank you so much for your time and I really enjoyed talking to you.